Hi, welcome to the second installment of Perceptual Optics. Today we are looking at voxel theater. Here, for example, is a 100 by 100 by 100 unit voxel theater, so it would be 1 million subdivisions. We're going to look at some definitions. What are pixels and voxels? We will look at visual paths through the brain. In other words, there's the primary one that goes through the thalamus to the visual cortex and provides us with conscious sight. There's also a path that goes to the pretectal nucleus and the Edinger-Westphal nucleus and controls your focus and your aperture size of your eyes. There's a superior colliculus path that is going to control saccade directions and amounts. There's also a path that goes to the suprachiasmatic nucleus to control circadian rhythms. We'll look at some requirements from experience. For example, we can open just our left eye or just our right eye and examine differences in the visual field and make some interesting observations there. We can take a look at how many voxels humans are capable of discriminating within conscious experience. We will look at how stereoscopic vision combines the left and the right eye's contributions. We'll look at computing and representing depth and vision as a single point versus as a whole field that you look at. What is a pixel? Pixel, short for picture element, is the smallest unit of an image that can be displayed or represented on the screen. It is a tiny square that contains a specific color and brightness value. While many pixels are arranged together in a grid, they form an image. The resolution of an image is determined by the number of pixels in the grid. The more pixels, the higher the resolution, the more detail you can see. Of course, you'll have pixels in things like your smartphone. What is a voxel? A voxel, or volume element, is the 3D equivalent of a pixel. So not only does it have width and height, it also has depth. You can think of this like in Star Wars, the R2-D2 hologram message from Leia had some resolution of voxels. So here we have a hand made out of voxels, or a voxelated hand. Obviously it's not as smooth as the hand over here. So even though they both could be considered made out of voxels, this would have a high resolution, this one on the right would have a low resolution. And so the physical world, even though you could think of it as being made up of voxels, it has an extremely high resolution. So down to the Heisenberg uncertainty principle kind of level, or the Planck minimum distance kind of level, we're going to have that kind of resolution in the physical world. So when I say not voxelated, I mean it's high resolution. It still has voxels, but it's high resolution. Perceptual world, on the other hand, also composed of voxels. It's going to be a lot lower resolution, so a lot more voxelated, but it's still so high resolution that we don't notice it. So for example, a human can discern a single arc minute. So if you hold your pinky out at arm's length, the fingernail will be about 2 degrees of theta. So if you divide that in half, half of your pinky will be a single degree. If you then take that and divide by 60, you have 1 arc minute. If you were to take that 1 arc minute and divide it into another 60 times, you'd have 1 arc second. So humans can discern 1 arc minute, and in lab settings, they may be able to get down to as precise as 2 to 6 arc seconds, which is tremendous resolution, but still way less than is available in the physical world. You could consider the physical world as divided into voxels or if you take an image with a camera and print the image out, you could think of the image as having a certain number of pixels. You could think of the screen of your phone having a certain number of pixels. So it's just a useful way to divide the physical world into units. The perceptual realm, which is the brain's schema, the brain's representation of the physical world or of imagination, also could be subdivided into pixels or voxels. In other words, the schema itself would be divided into voxels, but the retinal image prior to conversion to the 3D could be thought of as 2D and therefore pixels. As a recap, both the physical world and the perceptual realm can be subdivided into voxels, and under certain circumstances you can consider a perspective image of either of those that can be divided into pixels. Okay, the visual pathways, there's 4 primary pathways. Depicted here is the cortical path, which is the pathway for conscious sight. And the light enters the eyeball, goes through the optic nerve, through the optic chiasm, where part of it decussates or crosses to the contralateral side, but some of the fibers stay on the ipsilateral side. They go to these 2 lateral geniculate nuclei, one on the left, one on the right, and span into these optic radiation fibers that come to the visual cortex here in the back. There are also 3 other main paths. The pretectal path handles accommodation, or in other words, it's like the focus button of your camera. You can change what depth you want to focus to, and it also controls pupil size, which would be like the aperture of a camera. The path going to the superior colliculus controls saccade direction and amount, so like where you want to turn your head and eyes to look next. That's going to be controlled by the superior colliculus. And then you have this path going to the suprachiasmatic nucleus, which is in the hypothalamus, and it in turn will then send messages to the pineal gland to release melatonin and help you go to sleep when things get dark at nighttime. The main path for sight for us is going to be this first primary visual path. It's responsible for conscious sight perception. And like I said, it goes through the lateral geniculate nucleus and the thalamus, which is somewhat of a relay station. And then it goes to visual cortex V1, primary visual cortex, in the occipital lobe on the back of your head. And from there, it splits into a what and a where visual stream. And from those regions, it can go up to the prefrontal cortex, and then to the premotor, and then to the motor cortex, and actually drive your response to visual stimuli. The pretectal path is going to handle lens accommodation, which is focus distance, and pupil constriction, which is like aperture. And the visual information from the retina goes to the pretectal nucleus in the midbrain, processes light intensity and depth information. For lens accommodation, which is focus, pretectal nucleus sends signals to Edinger-Westphal, which in turn stimulates certain muscles to adjust the shape of the lens for optimal focus. In other words, if you're looking at infinity, it's going to stretch the lens out so that it has less focal power. And for things that are close up, it's going to relax it so that the lens becomes more like a sphere and has more refractive power, focusing power, so that you can focus on things that are closer up. For pupil constriction, the pretectal nucleus also communicates with Edinger-Westphal to control the sphincter pupillae muscles, resulting in pupillary constriction under bright light conditions. Superior colliculus pathway is involved in controlling saccadic eye movements and head direction movements. So The retina is going to capture visual information, send signals to superior colliculus in the midbrain, which plays a role in coordinating these movements. Also, the visual cortex can cause signals to be sent to the superior colliculus to drive movement. Of course, it would have to communicate first with frontal eye fields, supplementary eye fields, which are in the frontal lobe. But basically, the cortex can drive the movement of the eyes, but also if you see some dangerous stimuli or some interesting stimuli, your superior colliculus may turn your eyes and your head towards it involuntarily. Of course, you'll still be conscious that you're turning your head, but it was just like, oh, something startling, you just automatically turn your head and eyes there to look at it. Same thing with sound. The inferior colliculus is going to do the same thing, possibly in communication with the superior colliculus. If you hear some sound that's startling or something, you probably involuntarily turn your head that way to look at it. So the superior colliculus, we'll get into the layout of it later. There's also these other things, paramedian, pontine, reticular formation, all this other stuff that we'll look more in depth on this later. The suprachiasmatic nucleus path for circadian rhythms, the pathway from the eye to the brain region that regulates sleep at lower light levels, involves specialized photosensitive retinal ganglion cells that detect light levels, but it's not used for like a conscious image or anything like that. It's just the light level. It sends the information to the hypothalamus and specifically to the suprachiasmatic nucleus. And at the right time, when it turns dark, it's going to send signals to the pineal gland to release melatonin, which is going to regulate sleep cycles and make you sleepy and want to go to sleep. This is an image of the eye. So you have light coming through the cornea, pupil, lens, the eyeball itself. It's going to hit the retina on the back. You have the macula, and within that you have the smaller foveal zone. You have the optic nerve, which is where your blind spot is. It's where all the fibers come through and go to the brain. This image just shows you your cones, which are colored, red, blue, green, and your rods, which are for night vision and motion, and probably a little bit of other stuff like light-dark contrast. They get overexposed pretty easily, so a lot of the time they're kind of overexposed. This shows you that as light comes from the bottom in this case and goes up, it's going to have to first go through all these layers of neurons, and then it's going to hit the back right here where the cones and rods are at, and it's going to transform the form of vitamin A from cis to trans or trans to cis. It's a conformational change that has an effect on basically the electrical signaling of the cell. And actually, when it's dark, these cells are firing, and when they receive light, they stop firing. But there's another cell later in the path that converts that to the opposite, so that when you have light, you have firing, and when you have darkness, there's a cessation of firing. You got bipolar cells, you have amacrine cells, you have also horizontal cells. So this is a little bit of like kind of brain processing going on in the retina, actually in the eyeball. And then once you get to the retinal ganglion cells, these are the last step in the eyeball, and it's going to send the signal via these axons through the optic nerve. These are microscope images I took of a slide that had actual retina on it. And in this case, as they were mounting the specimen on the slide, the retina came detached from the pigment epithelium. So the pigment epithelium is where the basically vitamin A or some form thereof is kept, and it gets recycled back through the rods and cones. So it goes from one conformation to another, and then it has to get recycled out to the pigment epithelium and then back to the cones and rods after it's been converted back. that kind of thing. The cones and rods you can see right here. Not a lot to look at, but that's them. You've got choroid, you've got sclera, you have these layers like outer nuclear layer, outer plexiform layer, internuclear, inner plexiform, and then right here these little purple dots are the ganglion cell layer. So this is the last step in processing, and then the axons of these guys actually exit the eyeball and pass through optic chiasm and go on their way to lateral geniculate nucleus. So this shows you some more of these paths. So this one here is the pretectal on the right. So the signals are coming here, and then here's Edinger-Westphal, and then it goes back to the eyeball. And I believe both of these send signals also to lateral geniculate nucleus and pulvinar, and then on to the visual cortex. So the cortex knows about any instantaneous degree of focus or aperture size. On the left is just another depiction of the mostly of the primary visual path, but you also see the suprachiasmatic nucleus there. See if they got anything else here. I think that's about it. So you've got the you got these fibers coming along the optic tract. This is the optic chiasm right here, and lateral geniculate nucleus here and here. And basically the left visual field comes over to the right hemisphere, and the right visual field comes to the left visual cortex. And so part of the fibers are going to stay on the ipsilateral side, and part of them are going to decussate and cross to the contralateral side. This is showing you the lateral geniculate nucleus, and as you can see, it's got these 6 layers, and part of the region is going to be monocular. The other part is going to be binocular, and you have a layering of eyeballs too. So like you've got input from left and then right and then left, and there's a couple of layers where it's both the same eye and then it flips again. So it's a little bit unusual, but you also have parvocellular, magnocellular, and in between you have coniocellular. And the parvocellular is going to be primarily responsible for processing information on color, fine detail, spatial frequency. This layer receives input from the midget retinal ganglion cells, which have small receptive fields and are specialized for processing high-resolution visual information. Magnocellular layer is primarily responsible for processing information about motion, spatial orientation, and depth. This layer receives input from the parasol retinal ganglion cells, which have larger receptive fields and are specialized for processing low-resolution visual information. And the coniocellular layer in between is the smallest and least well understood of the 3 layers. It is thought to play a role in processing color and other visual features, but its precise functions are still not fully understood. So this is an image of the lateral geniculate nucleus here, and I believe this is layer 1 on the center, and going outward with 2, 3, 4, 5, and 6. You can also see, I think this would be putamen, I think basal ganglia, definitely hippocampus right here, coming out of the page and going back in, so you see a little bit of hippocampus up there, cerebellum down here. And this is an excellent resource here, braininfo.org. You can go through slices of the brain, I think in all 3 directions, like sagittal, coronal, and transverse, and look at it at varying resolution and things like that. So it's a very cool little resource. These are 4 visual pathways. Just to recap, the pretectal path for pupillary constriction, in other words, aperture size, lens accommodation, in other words, focal distance, and you also have the superior colliculus path, which is for guiding the direction and amount or distance of saccades, and also somewhat for your head direction, moving it. The suprachiasmatic nucleus path is going to handle circadian rhythms, and finally the visual cortex path is eventually going to lead to conscious perception of the visual field. Frontal eye fields will control voluntary movement of eyes. That's in the frontal lobe. Supplementary eye fields, also in the frontal lobe, help maintain gaze when turning head. For example, if I am focused on my index finger and I turn my head either way, my eyes are going to counter-rotate so that my gaze stays fixed on the index finger. If you are a runner and you're about to start a race and they have a red light that is going to turn green and trigger when you should start the race, you'll want to start as soon as possible. Now, when the light turns green, the visual input comes into your eye. The retinal ganglion cells are going to see the information 20 milliseconds later. The lateral geniculate nucleus will see the information at 50 milliseconds. Primary visual cortex, 70 milliseconds. The end of the what pathway and where pathway, 110 milliseconds. Prefrontal cortex will see it at 140. Premotor will see it at 170. And there's also the motor cortex, which I think was 210 milliseconds, covered up by the video. And then the motor response when the leg muscles engage is going to be 250 milliseconds. So a quarter of a second later, you actually respond and start running. And these numbers should be pretty close because there was actually a paper and a study done, and they tried to extrapolate over for humans to give accurate numbers here. All right, so requirements from experience, we see a seamless merger of left and right visual fields, even though the left field is going to the right visual cortex and vice versa. So somehow these are having to be merged together very, very well. Possibly even the schema, the real world schema, is created completely after all the processing is done. That's a possibility. But however it handles it, we can definitely see that there's no line in the middle between the left and right hemifields. So the information is joined together. And it's joined even better than the left and right eyes are joined. So the left and right hemifields are joined together more seamlessly than the left and right eyes are joined together. So that's pretty interesting. A single eye is able to see in 3D. In other words, when I look at this edge of the bed behind me, I don't see it as being in this fronto-parallel plane. I perceive it as actually being in the depths of going that way. So, and that's even with just one eye open. I still perceive the 3D direction correctly. So everything is seen in 3D, even if it's just with one eye, just monocular. Having two eyes does help us to have extra depth resolution, but a single eye still sees in 3D, so that's very critical to understand. The left eye voxel theater is laid over the right eye voxel theater. In other words, the whole 3D scene that the left eye or the right eye sees, when you have both eyes open, they get overlaid in the same 3D space, and only your focal point, whatever you're focused on, is going to get aligned spatially. That one point between the two 3D realms will be completely, perfectly spatially aligned. But from that point towards you, you'll have increasing crossed disparity, and from that point outwards, you'll have increasing uncrossed disparity. In other words, the two 3D scenes are overlaid, and they're shifted like that, so that the part that's closer to you gets increasingly crossed, and the part that's farther away from you gets increasingly pulled apart. And this is just a requirement of geometry, because we have two eyes that are separated by 2 inches. They're seeing a slightly different thing, and there's no way to merge both of what they are seeing except at one point. And so it's always the focal point. Whatever point you were looking at, that is the point that is going to be spatially aligned or fused between the two eyes, and no other point will be perfectly fused. An estimate for the number of voxels distinguishable by humans is around 7 trillion plus. We'll get to that. So as we were saying in the previous slide, there's only one point of the entire 3D scene of each eye that is spatially aligned, at least as far as perfect spatial alignment. And from that point outwards, you get increasing disparities. You get horizontal disparities because the two eyes are separated by 2 inches. You can also get vertical disparities. For example, if I hold a pen closer to my right eye and don't turn my nose, but just my eyes to look at the pen and focus on it, if I close my left eye, just look with the right eye, it's going to be taller than if I'm looking with just the left eye, because the pen is closer to my right eye, therefore it will take up more retinal height in the right eye than it does in the left eye. So that's a vertical disparity. And you can also see in this image that there's even rotational disparity and also size disparity. Like for this pixel here, the left eye is going to see an object or a feature right here as smaller than the right eye would see it, because it's closer to the right eye, and therefore it's going to take up more theta and more phi. So the size, but also the rotation, the pixel here will appear slightly rotated to the other eye. So there's just all these little funny disparities going on, and this focus point moves around. It can move inwards. You can have increased divergence and look at something closer. Like I can bring this pen closer to me, I can bring it farther away from me, I can bring this focal point anywhere in the 3D scene. Left, right, up, down, in, out. You could call this like a focal foveal voxel or a 3D cursor or a gaze point. Like it's this one point, and it's critical because this whole model here of what the eye sees is changing, and the disparities are changing in real time, very dynamically, based on where you're looking, how deep it is, how far it is, left and right, and up and down, and just all that stuff. So pretty important that this one point that we're looking at, and also these 2 points where the light is, all the light rays are coming to a point at the center of the right eye and the center of the left eye lens, at the center of the lens of each, and then going through and expanding again into an image, an inverted image on the back of the retina. So just to hammer this in, we have 3 very geometrically important points: the center of the left eye lens, the center of the right eye lens, and the focal point. Now, why are these 3 important? Well, this point is important because it's your focal distance, and it's the point you're looking at. And basically, if you know the 3D vector direction of this gaze direction of this eye and that eye, you know how far apart the eyes are, you have sufficient information just from that, just from eye directions, to calculate what the depth of this focal point is. Now, if the brain actually does it that way, I don't know, but geometrically it has enough information if it has a high resolution understanding of the exact 3D orientation of each gaze direction vector of the center of the visual field. If we were to imagine that we had a magical eye that could see in all directions right here at the center, up, down, behind, front, left, and right, then we could see in all directions simultaneously. And it would be possible for an animal to have an eye like this. And even though we can only see in this forwards direction, we can still turn our head around or turn our head up or down to see the entire sphere that is around us. So if you imagine that each of these little boxes was a pixel, then if you step the radius back slightly, you could create a voxel. And if you had several increments of radius, you could have a whole voxel theater, which we'll look at in the next slide. So here is that voxel theater fleshed out, at least with one radius shown. You could imagine that there's multiple radii, each as incrementally deep as they are wide and high. And this has 10,000 pixels, the little squares. And if you imagined 100 concentric layers going inwards, you could have 1 million voxels. Human vision can see at least 1 million times more resolution than this. So that's pretty powerful clarity. If you imagine that you are here at the origin looking to the right, and this is 20 feet away, 40 feet away, etc., and assuming that you can discriminate 1 arc minute of resolution, then this is the number of steps of depth discrimination cumulative that you would rack up as you start going outward from yourself. So you can see that most of your depth discrimination, or at least a sharp incline, is right here in this 20-foot radius around you. And as you add all the way to 200 feet, 10 times as much, you're only increasing from like 14,000 shells to 21,000 or something. So it's kind of like diminishing returns. And again, this is assuming a fairly low or fairly conservative assumption of 1 arc minute of resolution. Some lab results seem to indicate that humans may be able to resolve all the way down to 2 to 6 arc seconds, which would be a lot, which would basically mean that there's a whole lot more of these shells. So if we assume that humans can see 200 million pixels in a single sphere, in other words, a single radius, then we could basically turn the edge of the width or height so that it's now the depth. And so at any given radius outwards, the closer it is, the finer depth resolution you're going to have, so you'll have more shells or more steps of depth resolution discrimination. And as you get further away, like if something's 100 feet away, you might only be able to, the same 1 arc minute angle grows and becomes maybe an inch by there. And so if you turn an inch to the depth direction, okay, now you have much less depth discrimination than you did close up. So it's one of these things that it varies with the radius from you. But if we assume 35,000 steps of depth, I think that's pretty conservative in the 200 million pixels, then you could get to a number like 7 trillion voxels. So even though the eye can probably discern well over 1 trillion voxel locations, it's not like all those voxels are usually important to us. What's usually important is the surfacial skin of things. So like my shirt here is blocking you from seeing anything directly behind my shirt. So the voxels behind my shirt are pretty much not that useful. I mean, you may have some voxels to represent the kind of invisible backside of me, just so you have an understanding, okay, this is a human. So there may be a little bit of representation, but a lot of this space, like the air, it's not really that critical to remember all these voxels. What you want is the 3D boundary between an object, or the 3D surfacial skin that defines an object. For example, even the bed behind me, it's like, okay, this cover or this blanket blocks seeing any voxels underneath it. So what really kind of matters is just this very thin surface and its contours, the lines and the ripples and the texture and all that kind of thing. So it's just a few voxels that could be used to basically represent the shapes of things. And then the in-between, like air and stuff, is really not that critical. So it's kind of like the skins are important to save, or the surfacial boundaries. And even like in the visual cortex, a lot of what it's doing is edge detection in 2D. So in 2 dimensions, it's looking for the edges. Like if you got a square, it cares about the perimeter of the square, but it really doesn't care about the stuff inside as much. And I think it's the same thing with 3 dimensions. I think the brain cares about the surfacial boundaries that define an object and its shape, but not so much the innards or the air outside of the thing. If we make different assumptions for humans' theta resolution, we can come up with different numbers of total voxels that humans can discriminate. These two are in lab settings, so they're using, they're making it as easy as possible, and probably using very clear glasses or something that the person has the best, closest vision. However they do it, they use optimal settings, and supposedly have noted humans being able to discriminate down to 2 arc seconds or 6 arc seconds. But based on the assumption, you basically just say, okay, well, how many pixels per degree of theta is that? Okay, 60, okay, 600, 1800. And then how many pixels per square degree? Like, you know, you've got theta and you also got phi of height. So how many pixels per square degree? Okay, 3600, 360,000, 3 million. There's always 41,253 square degrees per sphere, so the whole area around you. So how many pixels can we see around us? Okay, 150 million, 15 billion, 130 billion, somewhere in there, you know. And if we then assume a depth range going from 5 centimeters, which is like 2.5 inches, all the way out to 3 kilometers, which is, you know, do you really want to see farther than that and have a lot of depth discrimination? It doesn't really matter beyond that point too much. So that's why I put that range. Okay, how many depth discrimination increments would you have? 38,000, 380,000, 1 million, and then how many voxels would that be? So that's just number of shells times total pixels per sphere. So you come up with 5 trillion, 5 quadrillion, 150 quadrillion. So using different assumptions, 1 arc minute, 6 arc seconds, 2 arc seconds, you can look at how many steps of depth discrimination you have cumulatively, starting from you being here and looking outwards, 500 meters, so on. Most of it is happening just right in the first little bit. So 0 to 5 meters, 0 to 10 meters, somewhere, you know, in that vicinity. So this, again, is assuming that you are standing here at the origin, looking to the right, 1 meter away, 2 meters away, assuming 2 arc seconds of resolution, which would be the very best that a human can see, then it would seem to indicate that within 5 meters you can see over 60 quadrillion voxels. And that's the entire sphere around you, not just the front direction, but also the back and up and down. Stereoscopic vision is often misunderstood as simply fusing the image from the left and right eye together. It actually doesn't really work like that. What actually happens is you have a full 3D realm attached to the left eye and a full 3D realm attached to the right eye. What you merge is only the focal point. So whatever object you are looking at, it's a point on that object where the central gaze vector of each eye is merging together, and that point will be spatially aligned between the two 3D realms. The rest of the 3D realm, they will be crossed, increasing cross disparity coming towards you and increasing uncrossed disparity going away from the focal point. Let's see if this adds anything here. Yes, okay, so there's a vertical axis. So if I'm looking at this pen, let's see here. All right, so if I'm looking at the tip of the pen, the left and right eye, they're 3D voxel theater. This tip of the pen will be aligned perfectly. But this thing that looks like 1 finger to you looks like 2 fingers to me, because I have 2 ghost copies, 1 here, 1 over here. But the truth is it's in the middle. That's because the 2 3D realms can't get completely overlaid, because the left and the right eye are separated by 2 inches. Therefore, geometrically, they cannot see the same exact thing. You know, if I had 2 fingers held up in front of me, the and I focus on this far one, my right eye will see the closer finger to the left of the farther finger, and my left eye will see the closer finger to the right of the farther finger. And again, to you, you just see the two fingers. But you can try that experiment for yourself, and you should see the same thing. The concept of a perceptual cyclopean eye being right in the middle of your other two eyes and looking out at the perceptual realm, and the perceptual realm being copied, the 3D realm being copied, the isometric 3D realm copied, and then kind of rotated. So if we start with these three crayons, red, yellow, and blue, and we make two complete 3D copies of this scene, and then rotate it such that the red crayon splits into two by crossing medially, and the blue crayon splits into two by splitting apart laterally, and then present it to a single cyclopean eye in the middle. This is pretty much, I think, geometrically equivalent to two eyes being separated by 2 inches and looking at a single scene, because these two eyes, again, are going to see two split copies of that, one copy of this, two split copies of that. So this is just showing the same thing, that your left eye is going to see the red crayon to the right of the focal yellow crown that it is looking at. It's going to see the blue crayon to the left. The right eye is going to see the opposite. It's going to see the red crayon to the left of the focal yellow crayon, and it's going to see the blue one to the right. So the left eye sees this, and the right eye sees that. And so how does the brain merge the two together? It does it like this. And you can test this for yourself. Hold your finger in front of your nose and look at a distant wall, and you'll see that your finger is in two copies, here and then over here. So they get split, and that's actually crossed disparity in that case. And so you see that the only object that is spatially aligned is the focal point, which you're focused on. The rest of it is going to experience a gradient of crossed or uncrossed disparity, or even the more complex disparities that we looked at, like rotational disparity, size disparity, things like that. So scientists believe that the brain uses disparity clues as perhaps the primary mechanism for precise depth understanding of objects. In other words, of turning the 2D retinal image into an understanding of where all those pixels should be cast to, or what radius should you assign to each of those pixel vectors. If you think about the image being, right there at the center of the avatar's lens, then you've got to cast it back out at all the directions, each pixel being its own unique vector direction pointing away from the center of the lens. If you assign just simply a radius to each one of those pixels, you can reconstitute an isometric representation of the 3D scene. So however the brain exactly does it, this seems to be one of the main tools, basically looking at horizontal disparities and maybe even more so vertical disparities. And we already kind of did that experiment where we held something in front of the right eye and saw that it could be look taller to the right eye than it does to the left. All right, so this slide is your payoff. This tells you all your monocular depth clues and binocular depth clues. So monocular is just like one eye, and this is a good reference, by the way, Ian Howard, Perceiving in Depth, and it talks about stereopsis and all of these things in great detail. But linear perspective, again, is just that as a pin with an absolute height moves away from me, it gets smaller in terms of phi, and how much height it takes up on my retina, even though it's absolutely staying the same size. So you can then use these clues of, okay, I know what size the pin is, how much space is it currently taking up on the retina, therefore it must be this certain distance away. Right? That's linear perspective. Occlusion is like one hand is occluding the other, therefore this hand must be behind this one. Texture gradient is if I held a piece of carpet here and the texture has a lower spatial frequency perceptually close up, because like 1 degree of horizontal theta may see only 3 plugs of carpet, whereas at the far end 1 degree of theta is now able to see 5 plugs of carpet. So it has a higher spatial frequency farther away than up close, and so you can use those clues of the change in spatial frequency of the texture to orient this 3D plane and understand the depth of that plane. I forgot to mention that occlusion is also called interposition. We talked about texture, gradient, spatial frequency. Focal power, or accommodation of the lens, can help you. The more spherical the lens is, the closer the object must be. Degree of divergence between the eyes. As I bring this pen closer to me and continue focusing on it, my eyes get increasingly more converged together. Motion parallax is if I look behind me and move laterally, I'll see that the bed moves faster than the piano. So, like for example, as I moved laterally, I could see that the bed was moving at a certain velocity and the piano was changing at a slower velocity, and based on the difference, I could calculate the depth between the two. Motion parallax is a very powerful tool and can even be monocular. Even with just one eye open, you can still do that and see a lot of detail of depth. Proprioception is just like kind of knowing where your body is, can provide a little bit of extra help. Binocular depth cues, stereo disparity that we've already talked about, horizontal disparity, vertical disparity, size disparity, maybe even rotational disparity could be used. So these clues are very powerful. I think they even said that there are binocular cells in V1 and maybe some other spots that actually are doing some calculation to determine depth based off of disparity. You can even have saccadic disparities between the eyes. In other words, you've got some fly buzzing around in 3D space. The eyes are having to, each saccade, the eyes are having to move a differential amount. And so this difference can be used to help you understand depth. Gaze direction vectors. If you knew exactly the orientation of the central ray coming out of your left eye and your right eye, then you would know exactly where they're meeting up. In other words, if I'm focusing on the tip of this point, the tip of this pen, just knowing the separation distance of the eyes, the head direction, the eye direction, and then the central ray coming out of each eye, I would have enough information to calculate exactly the depth of this voxel of this tip of this pen. Now, whether the brain does that or not, I don't know, but that's what that is there for. You could have disparities of accommodation. For example, I'm holding this pen closer to my right eye, therefore the right eye lens is more spherical than the left eye, because the left eye, the pen is further away, it does not have to focus as hard, therefore the left eye lens will be flatter. So the disparity of accommodation or focus could be used as a depth clue. So if you were a fish, you would have two eyes on the side of your head like this, and they would be pointing outwards, right? And then if you wanted to saccade these eyes, you could look in a forward direction, a backward direction. You could also look upwards or downwards, okay? And then humans have something kind of similar, but our eyes are here, and they're looking this way. So now we can look, again, up, down, but we can also look nasal or lateral. And so these are kind of the directions that are encoded by our superior colliculus. Fish don't have a superior colliculus, but they do have a tectal nucleus. So, and basically how this works is that, as I understand it, the superior colliculus is laid out such that the center neuron represents your focal gaze direction. So like the very center ray that's coming out of each eye, that is the center of your vision. And then on this superior colliculus map, then you can go nasal, you can go lateral, you can go up and down. So basically, whatever neuron fires, if a neuron close to the top of the superior colliculus fires, then that means, oh, bring your gaze direction upwards. And if it's just a little bit up, okay, just bring your gaze direction up a little. If it's over to the side, it's going to bring the gaze direction over that way. So it basically fires to move the gaze direction. Once the saccade settles at the new location, it updates that new location to be the new center of your superior colliculus, that neuron being responsible for that. And then you can refire in any of these other directions to move the eye from there. So it's kind of like this relative coordinate system that moves with and is always centered on your current center of view. So I think that's pretty interesting. But again, the superior colliculus handles that. Pretectal nucleus is going to handle lens accommodation information, such as, in other words, the focus and the pupil constriction. So this information passes to the eyeball, but it also goes to the lateral geniculate nucleus of the pulvinar of the thalamus. It goes to the visual cortex. And the visual cortex, well, we'll just say that the cortex, because the frontal eye fields and the supplementary eye fields, can send signals to superior colliculus to move the eyes, send signals to the pretectal to adjust the focus, or to adjust the, I'm not sure if it can adjust the pupil size, but it can definitely adjust the focus consciously. You may not have conscious control over pupil size, but the pretectal nucleus, based on the light information that's coming in, is going to adjust pupil size. So yeah, all that just to say that the cortex definitely has information on current focal depth and the movements of the eyes. Maybe not the absolute 3D vector orientation of the eye in some kind of absolute Euclidean space, but at least the relative motions it can know, and so it can kind of build this relative space of understanding of kind of where the eye is going to next and where it was at and that sort of thing. All right, so at this point we enter speculative territory, talking about how the brain actually represents 3D space. So one theory is volumetric representation, that there is basically perceptual space and every little voxel within it has a unique address one way or another. And one way that it could have a unique address is if a cortical column learned a unique ensemble of its neurons to encode that point. And then the idea is that the horizontal connections in layer 2, 3, or layer 5 of the cortex could communicate with other neighboring columns and eventually with the whole brain to make sure that there's an understanding of alignment between everybody's perceptual space, so that it's just one shared perceptual space. And if any column says, hey, I'm talking about this point in perceptual space, then every column understands it. So something like this needs to happen, I think, because if you think about it, all these columns up here, like somatosensory, is getting all these receptive fields. Like there's a receptive field for touching my tip of my left index finger. There's a column somewhere in the right somatosensory cortex dedicated that has a receptive field of paying attention when it's getting touched right there. Okay, but the thing is, it doesn't know if it's right here or right here or right here. See, this index finger can move to any of a trillion different places within perceptual space, and the brain really, really, really needs to understand where in perceptual space is the finger. It's not enough just to know, okay, it's this spot on the finger. I need to know that it's here and not here or here. And I need to know that not just for the tip of my index finger, but for every point on all of my skin, of my whole body. If I don't have that, I really don't have proprioception. The whole thing in proprioception is understanding location within perceptual space. So and that's somatosensory, but it could go for visual too, because visual cortex, it's got neurons that respond to certain amounts of theta or phi deviation from the center gaze point, and they're always responding to a certain amount of deviation from the center gaze point. Well, how do you know whether your head and eyes are looking here or here? How does it know whether the voxel that you're focused on is right here or right here? It could be in one of a trillion different places in perceptual space around you. So once again, it critically needs to understand perceptual space and where the receptive field currently is in perceptual space. Same thing for audio. It needs to know location. So that's pretty critical. I think that's a good idea. There's also object-centered representation. In other words, I see this cup. I can look at it from here. I'm forming these 2D images, and then I can combine those to get the 3D object. Supposedly, they even have some evidence from neurophysiological studies on that. Maybe, there's the structural description representation that scientists tend to like, of like, okay, you got these small features like little lines or curves or surface patches, and then as you climb the hierarchy, you assemble larger and larger items till you get up to like a full item. So, all right, so now we get to my idea on how the brain might do it. And this is like a refined bank theory, or you could call it like a transformable volumetric representation. So it's kind of like the volumetric thing of the last slide, but instead of having this perceptual space be fixed, what if we were able to, rotate it? Because you can imagine this cup, and you can close your eyes and imagine rotating it in the old direction, and I can control my motor commands to actually rotate the cup based on what I'm imagining in my imagination schema. So it seems to make a lot of sense to me that this space should be rotatable, scalable, because again, as this coffee cup gets closer to me, it is larger in perceptual space as far as the theta and the phi 2D is concerned. So it gets further away, it is smaller. So, and it could be at different locations, right? It could be here, it's hitting different neurons than if it's over here, even though it's the same size still. So you need to have invariance, shape invariance, regardless of what viewpoint or view location that you are at. It needs to be able to, the brain needs to be able to recognize that it's a coffee cup, it's a mug. So I think this is a very powerful idea here, that the brain has this perceptual space like a cube, and it has mappable voxels within it, and then it can rotate the thing, scale the thing, even translate it within the real-world schema. So this space out here is what I call real-world schema. So it's a representation made by the brain of the input coming from the physical world. So I don't think this is the physical world. I think it's a very good dashboard for it and a representation, but I don't think it is the essence of the physical world. I think this is completely the schema created by the brain. And so we'll get to that more later. But yeah, so that's kind of my idea there. And next slide we'll look at it a little bit further. All right, so not only is this proposed volume transformable, rotatable, scalable, translatable within the real-world schema, I think it also has multiple instantiation by the brain simultaneously. So I think there's one copy dedicated to representing the real-world schema, which is like, right now it's this room for me, which is like, it needs to be 15 foot wide, 15 foot long, 9 feet tall in order to represent everything in here. So it had to scale up to represent this space. If a car drives by, it needs to scale up immediately or very quickly to represent the car going past. Even though I can't see it, I still understand where it's going. So that would be one copy. Another copy would be dedicated to your imagination. For example, if you hear someone speaking or you hear a song, you're going to start generating some mental imagery, which usually happens somewhere in this space right here. So what is this, like 1/10 of the width of the real world schema, 1/10 of the length, 1/10 of the height. So it's usually like, what is that, 1/1000th of the volume? Because I mean, you can have mental imagery at other places, but generally it's going to be kind of close to your head in this general vicinity. It's not inside your head. It's kind of a little bit usually outside of the head, but this general vicinity. So that would be a whole instantiation or copy of the volumetric transformable representational space, and then I would postulate that there's a third copy that is used for the bank, which is the memory store of all 2D outlines and 3D surfaces, 3D shapes, skins, objects. So every shape that you've ever learned would be stored in this bank, and it's an inflexible structure. Unlike the other two, things don't move around inside of it. Now, you could save, like, your toe wiggling by, you could have like a really giant toe that, over the course of a second, it sequentially illuminates this skin, 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 so that it looks like motion, it looks like it's moving, but everything is still glued together and there wasn't any actual movement. And so that's what I'm saying with this bank structure. It's like a memory store of shapes, and everything inside it is fixed, it's glued, it's cohered together. And now you can take the whole thing and scale it up, rotate it within the real world schema, you can translate it. So, if I've learned this shape, then I'm saying that there's a bank that has this cup within it, and it can take it, translate it over here, rotate it to the right rotation, and say, oh yeah, I have familiarity of this thing because it's lining up. And then you could also use that as a system of like triggering other like things. So if there's another cup, then I have kind of an understanding of cups because they're all kind of the same shape, more or less. This chart here is just showing that there's levels of size within the brain. So you've got the whole brain, you've got regions within, like visual cortex 1, V2, V4. You can pull out one region and look at all the cortical columns within it. You can pull out a single column and look at the neurons within it and the layers of the cortex, so like layer 1, layer 2, 3, which has a lot of horizontal connections, layer 4, where a lot of your sensory input comes to, layer 5/6, where it also has a little bit of horizontal connections. Layer 5 drives your motor neurons. Layer 6 connects with other parts of the cortex by going through the thalamus and then going back to the cortex. You can also look at an individual neuron and the axons that come and kind of summate and whether the neuron fires or not in response to certain specific instances. So you got all these layers of scale, of size happening in the brain, which makes it very complex. But if you look here, it says that the cortical column has 40,000 neurons. Layer 2, 3 has 10,000 neurons. If you only want to rely on a sparse amount of those neurons firing, I'm not sure if I'm using that word correctly, but if you only want 1% of the neurons firing at any given time, that would be 400 neurons. And if you want to have robustness such that even when only 20% of those are firing, then let's see, that'd be 400. What was that, like 80 neurons? So even with just that, you can still encode a tremendous amount of information. Because if you think about it like a JPEG, shooting in JPEG with 8 bits, like 8 binary digits, zeros or ones, you can store 256 combinations, patterns. If you just add a few more zeros and ones and have a 14-bit of data, now you're at a raw camera image, and you can store like 64,000 something combinations. So just adding a few extra digits, you can save an astronomical amount. So once you go from like 14 bits to 80 bits, that's a tremendous amount of capability, and I think that it's able to represent well over 100 quadrillion voxels, if in fact that is what these ensembles of neurons are for. So that's just an interesting possibility. we typically think of vision as this large field, and we definitely have that component. But if you stop and think about it, a lot of vision is all about a point. So if I'm like looking like right at my eye or right at my nose, that's what I'm looking at. Or if I'm looking at the tip of this pen here, let's get it in the view. All right, if I'm looking at the tip of the pen, then that's what I'm kind of paying attention to. So believe it or not, a lot of vision is a very small area. So again, if you hold up your pinky fingernail, and let's say that that's 2 degrees theta by 2 degrees phi, then that's like, what, 4 square degrees. That is most of what you're looking at. And as you go about the world, you're moving your eyes so that the thing of interest is always falling on pretty much the same set of neurons. It's right there at the fovea center, and also on the at the back of your head at the visual cortex. The features of interest are pretty much falling on the same set of neurons. Now it does, I think, blow up the central vision so that you have more neurons dedicated to it, which is good. But it's falling on these same neurons, and the periphery neurons. I think that's probably why they're used a lot for motion detection and stuff, because we really don't pay a lot of attention to this stuff out here. It's like all these cones and rods and visual cortex, all these neurons, and we don't use that a whole lot. So the focal point is kind of a big deal, and more of a deal than really you've probably maybe considered. So this is just a little experiment. If I have one eye open and I focus on the tip of my right index finger, I can bring my focus to something in the background, like in this case my computer screen, or I can shift it back to my tip of my index finger. So in that case, I'm not moving left or right or up or down in terms of pixels. I'm just moving forwards or back in terms of voxels. So that's just kind of an interesting thing to kind of get your head around the idea of moving a point in 3D space, kind of like like your point of interest is like a 3D cursor, so to speak. So this is just a citation for this, and this paper here says that there's a dissociable spatial mapping for each eye. In other words, what that's saying is, hey, we really don't fuse the left and right eye together except at that one little point of focus. The rest of the 3D fields, or the 3D voxel theater that each eye sees, is experiencing cross disparities and uncross disparities. Therefore, there's a complete dissociable spatial mapping or 3D realm for each eye. Like there's the left eye, what it sees, and then there's the right eye, what it sees, and the two are overlaid, which have a little bit of monocular zone on the sides, but it's a lot of binocular zone, and for that binocular zone, there is no fusing. That's basically, I think, what he's saying. It's the focal point is the only spatial spot where the two are spatially aligned. So this paper is just illustrating that humans are continuously shifting their gaze point. He calls it a gaze point, which is that's a good term. I've called it focal foveal voxel, which is kind of a little bit lacking, or 3D cursor, which could be confusing. But your gaze point in three-dimensional space, it's more important than you might be led to believe. All right, so here we are looking at a housefly that's making some trek in 3D space, and our gaze is, we're saccading our eyes, but they're saccading differentially a different amount to keep up with the fly, but it's in 3D space, and the point of interest is kind of like a 3D cursor, or like a gaze point that's moving around in 3D space. And one reason that this is important is because I think the brain kind of starts with this point. It's a pretty critical point for building out the real world schema because it's like it's the merger. It's the point of interest. It's the point where there's fusing and spatial alignment between the two 3D voxel theaters of each eye. And finally, I know this has been a very long class, lots of things to think about, but this chart here is just giving you one last summation. We start with this star cluster. All the light rays are coming in to the pupil of the eye and then to the lens, and at the center of the lens, these rays are converging, not because of the power of the lens, but because of the concept of a pinhole, or just any camera or eyeball or anything. The light rays are coming from their various orientations, and they meet this point where they invert. But at that point, all the information of the 2D image is contained in a zero-dimensional point, which is very fascinating. So then the brain has to go, it has all these orientations saved here on the retina, and it has to start from the avatar's pupil, or lens rather, the center of its lens, and cast each one of these pixels back to the proper radial distance so that you can reconstitute the 3D isometric representation of the outside world from this. Basically, it's 2D information, but it's right here. It's in a zero-dimensional spot. But the brain has this information, and somehow it casts all these light rays back to their originating cause distance to build the model. And then, once you've got this 3D model or voxel theater for each eye, with the eye being at the very center, looking outwards, and of course we don't see the whole sphere, but it's just illustrative of the fact that we can turn our head and our eyes, and we can, over time, see the whole sphere, even though we can't see it instantaneously. So you've got this voxel theater for each eye. You have to add in, quote, the avatar, in other words, your body. Like, if I'm looking, I may not see, if I'm looking up, I don't see the rest of my body, but proprioception and somatosensory cortex has to still add it in. I still have understanding of where it is. And then if I'm happen to be also looking at the avatar, then that's extra visual reinforcement of what the proprioception information was giving me. So you have to add, quote, add the avatar, add sound locations, add touch locations, consider coloring. And once you do that, then we'll explore in the next video how we can add the bank to, that's the structure here, to basically record new shapes that are of interest. I would think that even episodic memory somehow is stored by, adding it to this bank. So once you have the bank, you can, there may be some interplay between that and the actual frames of consciousness. So if you look at the fact that consciousness is discontinuous at a fine temporal resolution, then these are specific orientations that the bank is at, and it's moving around. Like if I have my phone here, I could bring my attention to the phone, and the bank would be here with the cylinder on the phone, and then I might lean back a little in my chair, and the bank could come over here transiently, paint the focus on my body and the feeling of leaning back in the chair, and then move back to the phone, and then maybe there's a noise over here, and it's like the bank moves over there, prints that, records that, comes back over here. So it's basically like whatever you are focusing, whatever is within your purview of focus, that is what is recorded to memory, and it's done so with the bank. And what the relationship between memory and consciousness, like if there's more consciousness than is recorded, or whether it's an eidetic amount, I don't know. But obviously, once you try to record something, you don't have perfect recall of it. But that's kind of a complex question. But basically, this just gives you a little overview of what we talked about. And we'll see you in the next class. Thank you.